nice to see you again. Let's discuss evolution and development, shall we? Closely related species of frogs can have different patterns of development. One frog goes from fertilized egg to adult frog with no intermediate stage. The sister species has an extra developmental stage, neatly slipped in between an early development and the formation of limbs. Research findings are accentuating the biological puzzle that many developmental genes are highly conserved, and a tremendous diversity of life shares this basic toolkit of developmental genes. In this lecture, I elaborate on the emerging field of evolution of development. Heritable changes in development are the result of changes in genes or gene expression that produce a different phenotype. Phenotypic diversity could either result from changes in protein coding regions of many different genes across species, or could be explained by how a much smaller set of genes is deployed and regulated. In some cases, changes in the protein coding region have been implicated in novel phenotypes. In other cases, a set of genes conserved across species appears to be responsible for the basic body plan of organisms, with changes in regulation of gene expression, accounting for phenotypic differences. DNA is a polynucleotide. It's made up of lots of nucleotides joined. Each nucleotide is made from a pentose sugar with 5 carbon atoms, a phosphate group, and a nitrogenous base. The sugar in DNA nucleotides is a deoxyribose sugar. Each nucleotide has the same sugar and phosphate. The base on each nucleotide can vary though. There are 4 possible bases: adenine, thymine, cytosine, and guanine. DNA nucleotides join to form polynucleotide strands. The nucleotides join up between the phosphate groups of one nucleotide and the other, sugar of another, creating a sugar-phosphate backbone. 2 DNA polynucleotide strands join by hydrogen bonding between the bases. Each base can only join with one partner. This is called a specific base pairing. Adenine always pairs with thymine, and guanine always pairs with cytosine. The 2 strands wind up to form a DNA double helix. Genes are sections of DNA. They are found on chromosomes. Genes code for proteins, polypeptides. They contain the instructions to make them. Proteins are made from amino acids. Different proteins have a different number and order of amino acids. It is the order of nucleotide bases in a gene that determines the order of amino acids in a particular protein. Each amino acid is coded for by a sequence of 3 bases, called a triplet in a gene. A DNA triplet is also called a base triplet or codon. Now, different sequences of bases code for different amino acids. This is the genetic code. DNA molecules are found in the nucleus of the cells, but the organelles for protein synthesis, that is ribosomes, are found in the cytoplasm. DNA is too large to move out of the nucleus, so a section is copied into RNA. This process is called transcription. The DNA leaves the nucleus and joins with a ribosome in the cytoplasm, where it can be used to synthesize a protein. This process is called translation. Transcription starts from RNA polymerase, an enzyme, attaches to the DNA double helix at the beginning of a gene. The hydrogen bonds between the 2 DNA strands in the gene break, separating the strands, and the DNA molecules uncoil at that point. One of the strands is then used as a template to make an mRNA copy. The RNA polymerase lines up free RNA nucleotides alongside the template strand. Specific base pairing means that the mRNA strands ends up being a complementary copy of the DNA template strand. Once the RNA nucleotides have paired up with their specific bases on the DNA strand, they are joined together, forming an mRNA molecule. The RNA polymerase moves along the DNA, separating the strands and assembling the mRNA strand. The hydrogen bonds between the uncoiled strand of DNA reform once the RNA polymerase has passed by and the strands coil back into the double helix. The RNA polymerase reaches a particular sequence of DNA called a stop signal. It stops making mRNA and detaches from the DNA. The mRNA moves out of the nucleus through a nuclear pore and attaches to a ribosome in the cytoplasm, where the next stage of protein synthesis takes place. Not all the genes in a cell are expressed, transcribed, and used to make protein. Because different genes are expressed, different proteins are made, and these proteins modify the cell. They determine the cell structure and control all cell processes. Transcription factors and genes involved in signaling pathways are responsible for coordination of development. Even subtle changes in a signaling pathway can alter the enzyme that is activated or repressed, the transcription factor that is activated or repressed, or the activation or repression of gene expression. Any of these changes can have dramatic effects on the development of an organism. Hox, homeobox genes, appeared before the divergence of plants and animals. In animals, they establish body plans. These genes code for proteins, with a highly conserved homeodomain that binds to the regulatory regions of other genes to activate or repress these genes' expressions. Hox genes specify when and where genes are expressed. Understanding how development evolves requires integration of knowledge about genes, gene expression, development, and evolution. Either transcription factors or signaling molecules can be modified during evolution, changing the timing or position of gene expression, and, as a result, gene function. Alterations in timing of developmental events due to a genetic change are called heterochrony. Every so often, a novel phenotype emerges that persists because of increased fitness. Alterations in the spatial pattern of gene expression can result in homeosis. The transformation of the hindwings into minute halteres with a sensory function is the outstanding apomorphic feature of the Diptera. A four-winged Drosophila fly is an example of a change in gene expression pattern with a dramatic effect on morphology. Mutations in 3 genes in the bithorax complex are required to produce this phenotype, which resembles more ancestral insects with 4 rather than 2 wings. The timing or location of gene expression can be modified in several ways, giving rise to heterochrony or homeosis. The coding sequence of a gene can contain multiple regions with the different functions. The DNA binding motifs exemplified by Hox genes could be altered so that they no longer bind to their target genes. As a result, that developmental pathway would cease to function. Alternatively, the modified transcription factor might bind to a different target and initiate a new sequence of developmental events. Signaling pathways help cells coordinate information about neighboring cells and the external environment that is essential for successful development. If the structure of a ligand changes, it may no longer bind to its target receptor. It could bind to a different receptor or no receptor at all. If, because of a genetic change, a receptor is produced in a different cell type, a homeotic phenotype may appear. And, as mentioned earlier, small changes in signaling molecules can alter their targets. Wild cabbage, kale, tree kale, red cabbage, green cabbage, Brussels sprouts, broccoli and cauliflower are all members of the same species. Some flower early, some late. Curiously, these plants with such different appearances are very closely related. A point mutation that converted an amino acid coding region into a stop codon resulted in the extensive reproduction branching pattern that was artificially selected for in 2 crop plants that are both Brassica alteracea. Homoplastic structures, also known as analogous structures, have the same or similar functions, but arose independently. Unlike homologous structures that arose once from a common ancestor, phylogenies reveal convergent events. But the origin of the convergence may not be easily understood. In many cases, different developmental pathways have been modified, as is the case with spots on a butterfly wings. Insect wings have patterns that can protect them from predation. The origin of these patterns are best explained by co-option, the recruitment of existing regulatory programs for new functions. Distal less is one of the genes co-opted for butterfly spot development. Limb development in insects and arthropods requires distal less, but the expression of this gene also predicts where spots will form on butterfly wings. Highly conserved genes can undergo small changes in the coding or regulatory regions that alter the place or time of gene expression and function, resulting in new body plans. Changes in transcription factors and signaling pathways are the most common source of new morphologies. Although most mutations are lethal, some confer a fitness advantage. These may consist of very small mutations, such as the change to a single codon, that have large effects on development and morphology. During the long course of evolution, genes have been co-opted for new functions. A change in the protein coding region of a transcription factor can change the genes it can bind to and regulate. A change in this regulatory region of a gene can change where or when that gene is expressed, which can lead to altered morphology. Homoplastic features have a similar function but different evolutionary origins and are examples of convergent evolution. Homologous features have the same evolutionary origins but may have a different function. I hope you've learned something from this lecture. I'll see you again soon.